Hello students, welcome to this video lecture series on biochemistry. I am your teacher Ashwini Kumar Nigam. In previous online lectures, we have studied all about the amino acids, their properties, classification, ionization pattern and physiological significances. Today we will learn about cysteine molecule, formation of peptide bond, types of peptides, nomenclature of peptides, and then nature and properties of peptide bond.
First of all, I shall explain you people about the cysteine, which is asked frequently in examinations. Okay, so there are two different words you must understand: cysteine and cysteine. Cysteine spelling I am giving you people. CYSTEINE is cysteine and CYSTINE is cysteine. And in previous lecture we have studied about the amino acid cysteine, which is one of the two amino acids which are containing sulfur. So sulfur containing amino acids are cysteine and methionine, but we have also seen that in cysteine there is free sulfhydryl group is present, which is lacking in methionine. The free sulfhydryl group present in cysteine can form disulfide bond, in such a way so that two cysteine molecules are combined through this disulfide bond to form a single molecule of cysteine. Cysteine thus is a dimer of two cysteine molecules joined together by disulfide bond, and therefore cysteine is oxidized dimeric form of cysteine. Free sulfhydryl group present in two cysteine molecules are oxidized to form disulfide bond of cysteine. So here you can see in the structure that free sulfhydryl groups in cysteine molecules are highlighted in yellow color, and which after the formation of disulfide bond form the cysteine molecule, and that disulfide bond in cysteine molecule is also highlighted in yellow color. So in this way cysteine molecule is reduced form, and after oxidation it is forming the oxidized form of the molecule, which is known as cysteine. And these two forms are interconvertible depending upon the physiological state of the body or presence or absence of the any reducing or oxidizing agents. Disulfide bonds play important role in folding and maintenance of three-dimensional structure of proteins.
Okay, now we will learn about the formation of peptide bonds. As we know that each and every amino acid has two main functional groups, an alpha amino group and a carboxyl group, which are connected to the central alpha carbon atom, and these functional groups are very important in the formation of peptide bonds to form a peptide or polypeptide chain. Now question arises how the peptide bond is formed, or how these amino and carboxyl groups are involved in the formation of peptide bond to form a particular polypeptide chain. So peptide bond is formed by removal of the elements of water from the alpha carboxyl group of one amino acid and from the alpha amino group of another amino acid molecule. Thus two amino acid molecules are covalently joined together through a substituted amide linkage called peptide bond to yield a dipeptide. If we talk about the two amino acid constituents, then resulting molecule will be known as dipeptide joined together by a covalent bond, which is known as peptide bond here. So how these two groups are involved? So you can see here that hydroxyl group of carboxyl group of one or first amino acid is reacted with the hydrogen of the, or you can say that will combine with the hydrogen of the amino group of the next amino acid to form a peptide bond, which is highlighted in yellow color here in this structure, and with the removal of the water molecule. Just because of the removal of the water molecule, the reaction is or you can say formation of peptide bond is a type of dehydration reaction, okay, because water molecule is released here. Now, why it is written as condensation reaction? Just because two amino acids are here condensed to form a dipeptide or to yield a dipeptide molecule. Okay. Now, we can see here a reverse reaction, okay, where it is written as hydrolysis or proteolysis of peptide bond. Okay. So, when peptide bond is formed, this is the dehydration or condensation reaction, but whenever this bond is broken down, the reaction is known as hydrolysis or proteolysis of peptide bond, which are occurring inside our digestive system. Okay. Digestive proteases such as pepsin, trypsin, chymotrypsins are those enzymes which are breaking these bond. In fact, these bonds are broken down by these types of enzymatic molecule to break down the whole larger molecule of the protein or polypeptide chain into the smaller oligopeptide chain, or to yield a final free amino acid which are absorbed at the enterocyte surface. Okay. Now, next slide is concerning with the properties and different types of terminals of the resulting peptide. Okay. So, we can see here there are two free terminals after the formation of the peptide or dipeptide or whatever polypeptide is formed. Okay. So, there are two free terminal, one is N terminal or amino terminal, that will be of the first amino acid where that particular group has not reacted with any of the carboxyl group of any other amino acid. So, you can say the amino terminal is the free terminal of the first amino acid, or you can say what will be the free terminal or free group of the first amino acid, that will be amino terminal. Now, you can see at the right side free C terminal or carboxy terminal, that is the terminal which is free and that is not reacted with the amino terminal of any other type of amino acid. So, you can have two types of free terminals in each and every type of peptide, oligopeptide or polypeptide, whatever, or you can say a whole in a whole protein molecule. So, there will be two free terminals, one is amino group, that will be called as N terminal, okay, which is in fact the free amino terminal of first amino acid, and another group is carboxyl terminal, which is in fact a free carboxyl terminal of last amino acid. Okay. Now, each constituent amino acids in a polypeptide or dipeptide or tripeptide after the formation of a peptide bond are known as residue or amino acid residues. Each amino acid component in a polypeptide chain is designated now as residue. Why it is called residue? Just because of the removal of elements of water during the formation of peptide bond. Okay. If you recall from the previous slide, hydroxyl group of the carboxyl group is removed from the first amino acid, which is reacting with the hydrogen of the amino group of the next amino acid to form a peptide bond with the release of the water. So, that amino acid is not in the form of complete structure after the formation of peptide bonds. So, those are residual structures or residual moieties after the formation of peptide. So, those amino acid constituents are known as amino acid residues. Technically, these are not complete amino acid structures, just because of the removal of some elements or some moieties during the condensation or dehydration reaction to form peptide bond. Now, question arises, where or in which cellular compartment this peptide bond is formed? In cytoplasm of the cell, peptide bond is formed by the action of peptidyl transferase enzyme on the ribosomal assembly governed by the genetic codes present on mRNA. So, you can say in simple language, peptidyl transferase enzymes are responsible for the formation of peptide bonds between amino acid molecules or between constituent amino acids. Now, after the formation of peptides, they are classified in different categories depending upon the number of amino acid residues in that peptide or polypeptide. So, the whole peptide, technically the whole peptides are categorized into two main categories, oligopeptides and polypeptides. So, oligopeptides are short chain polymers of few amino acids, means 2 to 9 or 10 or 11 amino acids joined through peptide bonds. So, whenever a particular short peptide is formed, that is recognized as or called as oligopeptides, and they can be dipeptide, tripeptide, tetrapeptide, pentapeptide, hexapeptide, heptapeptide, octapeptide, nonapeptide or decapeptide and so on, just depending upon the number of amino acids. So, one thing should be remembered here, one particular point should be noted, that a dipeptide word does not mean the presence of two peptide bonds. Okay, dipeptide means presence of two amino residues joined together one peptide bond. Okay, so there will be one peptide bond between two amino acid residues in a dipeptide. So, likewise in tripeptide there will be three amino acids with two peptide bonds, and tetrapeptide will be containing four amino acid residues which are bound together by three consecutive peptide bonds. Okay. So, whenever particular question is asked about the number of the amino acid or number of the peptide bonds present in a particular type of oligopeptide, you will write in such a manner that if you see a dipeptide in a question, you will write two amino acid joined together by one peptide bond. Okay, understood. So, next category is polypeptide. So, polypeptides are long chain polymers of several amino acids joined together through peptide bonds. Several means they may have as many as hundreds to thousand of amino acids. So, there will be 100 to 1000 amino acid residues in a polypeptide. So, you can say peptides are short or long chain polymers of L alpha amino acids joined together by peptide bonds. These are the technical classification, but in real field of the biochemistry, these terminologies are not going on. So, in general in biochemistry, the term peptide is exclusively used to denote short chain peptides, and the larger peptides are called directly polypeptides or protein. Okay. Now, after the formation of short peptides or oligopeptides, say for example that peptide is composed of 2 or 3 or more amino acid up to the 9 or 10 amino acid, they can be named based on the sequence of the peptide. Okay. So, for specific nomenclature of peptides, a certain rule is followed and that are to be described here. So, there are two very simple rules for the nomenclature of peptides. First is to replace I and E, last three letters of each amino acid which are towards the N terminal, and those I and E letters are replaced by YL. Okay. And second rule is not to replace I and E, means last three letters of amino acid by the YL, we will not replace just for the last amino acid or the amino acid situated at C terminus. Say for example, or you can say there are two examples of dipeptides. First is A, or you can say first is composed of glycine and alanine, means glycine is at N terminus and alanine is at C terminus, and there is also second dipeptide in which alanine is situated at N terminus and glycine is situated at G terminus. Although these two dipeptides are made up of two amino acids, those which are glycine and alanine, but the sequence is different. In first is glycine and then alanine, and in second dipeptide you will see alanine is first and glycine is second. So these two are different dipeptides. These are not similar dipeptides. These are not two same dipeptides. These are two different dipeptides. For the nomenclature, we will follow those rules here. So we will first replace I and E letters of the glycine, and then we will add YL. Okay, the glycine I and E, after the replacement it is known as glycyl. Okay, GLYCYL. ठीक, तो glycyl. Then there is next amino acid which is at the C terminus. We will not replace I and E here, so that will be written as whole alanine. The name of the dipeptide will be glycyl alanine, and likewise next peptide is known as alanyl glycine. Here ion of alanine which is present at the N terminus, or which is in fact here is the first amino acid. So following these two rules, we may further name these two tripeptides. So tripeptides are here, two tripeptides are here. One is composed of serine, glycine and lysine. Serine is at N terminus, glycine is in the middle of the sequence, and lysine is at C terminus. Then second type tripeptide is lysine, glycine, serine. Here lysine is at the N terminus, glycine as usual in middle of the sequence, and serine now here is at the C terminus. So following those two rules of the nomenclature of peptides, the name of the first tripeptide will be seryl glycyl lysine. On the other hand, the name of the next tripeptide will be lysyl glycyl serine. Okay, so these are few examples just to explain how will you give names of the peptides. Okay, understood? Okay, now we know very well that how peptide bond is formed, but after the formation of peptide bond, its nature or property becomes different. Why? Because two reactive groups of amino acids, means carboxyl groups and amino groups, are reacted together to form a peptide bond. But this simple bond is not so simple as we see it as single bond. Okay, this peptide bond seems a single bond, but it is showing a partial double bond character which is quite different from the any amide bond or any primary amine bond. Okay, how it is becoming different in nature, or how particular peptide bond is regarded or converted into partial double bond character showing a specific type of bond. So those basics or those chemical basics we will learn in this section. So just before the understanding basics of the partial double bond character and coplanarity of those atoms around the peptide bond, we must understand some of the basics of the peptide bond. So just after the formation of the peptide bond, there is also a appearance of peptide group. Okay, so that peptide group is consisting of carbonyl oxygen, carbonyl carbon, which are leftover part of the carboxyl group after the removal of the hydroxyl group. Okay, and second and the third and fourth atoms of the peptide group is peptide nitrogen and peptide hydrogen. So you can see here four specific atoms: carbonyl oxygen, carbonyl carbon, amide or peptide nitrogen and peptide hydrogen. All these four atoms constitute peptide group. Okay, and there is a peptide bond between the carbonyl carbon and amide or peptide nitrogen. So this CN bond is regarded as, is said to be a peptide bond. Oh, okay. So to understand the partial double bond character, we first target upon the lone pairs of the electrons which are present on the nitrogen and which are not involved in the any bond formation. So, those lone pairs of electrons are there on nitrogen, or you can say peptide nitrogen, and there is also oxygen, carbonyl oxygen, that is more electronegative than the peptide nitrogen. So, higher electronegativity of the oxygen will pull those lone pair of electrons on the nitrogen towards it, means electronegativity of oxygen is attracting the lone pairs of electrons from the nitrogen atom through the carbonyl carbon. So, you can say there is a sharing of lone pair of electrons between nitrogen and oxygen, or there is a sharing of lone pairs of electrons between peptide nitrogen and carbonyl oxygen through a carbonyl carbon. So, just because of this there is existence of resonance between the atoms. So, just because of the sharing of those electrons, there are three resonance structures you can see here. In first, there is a 100% single bond character in the peptide bond, then in the middle of the structure there is a partial double bond character just because of the sharing of the lone pairs of the electrons between nitrogen and oxygen, and then there is a development of pure double bond character in the peptide bond, and there is also development of positive charge on the peptide nitrogen and negative charge on the peptide or carbonyl oxygen. So, just because of the resonance stabilization, the middle of the structure is stabilized, therefore, there is a partial double bond character, or you can say if you measure the length of this peptide bond after the X-ray crystallography, you will find that the bond length is between single bond and the double bond, that is not purely single bond and that is also not purely double bond. So, that is why this nature of the peptide bond is known as partial double bond character of the peptide bond, and just because of this double bond character, rotation around this CN peptide bond is not possible, and that is why this peptide group is rigid, and all the atoms of the peptide group and around the peptide group, okay, they are coplanar structures. So, we will study coplanarity of these atoms around the peptide bond in the next section. So, we can see here just because of the no rotation around the peptide bond CN bond, there are 6 atoms which lie in the same plane, okay, or you can say there are 6 atoms that lie in the same rigid plane of the peptide unit. Okay. So, what are those atoms? So, first is carbonyl oxygen, then carbonyl carbon, then amide nitrogen or peptide nitrogen, then amide or peptide hydrogen, then C alpha N. C alpha N means C alpha of first or previous N terminus amino acid residue, and the last atom or sixth atom of this plain peptide unit is C alpha N plus 1, or you can say C alpha carbon of next amino acid towards the C terminus. Okay. So, these 6 atoms are in the same rigid plane of the peptide unit just because of the restriction of the rotation around the peptide bond. So, therefore, rotation is possible around only 2 bonds. What are those 2 bonds? C alpha and C bond. Okay, you can see here the rotation around the C alpha and C causing the dihedral angle which is known as psi angle, PSI, psi angle. Okay. Now, next bond around which rotation is possible is N to C alpha bond causing the dihedral or torsion angle known as phi angle, PHI, phi angle. Okay. So, rotation is only possible around C alpha C bond and N to C alpha bond, and rotation is not allowed around the peptide bond. And whenever there is formation of such type of the bond, the carbonyl oxygen and amide hydrogen or peptide hydrogen are always in trans position, not in cis position. In all sorts of naturally occurring peptide bonds, you will see carbonyl oxygen and amide hydrogen or peptide hydrogen are trans to each other. They are not lying just facing each other in cis position. There this type of torsion means this type of torsion angle, which is designated as omega angle, is sterically not possible. Okay. So, in all sorts of the peptides, you will see all those carbonyl oxygen and amide hydrogen are trans to each other, or you can say for all naturally occurring peptide, the omega torsion angle for the peptide bond, which is fixed angle, is always 180 degree, not the 0 degree, just because of the steric hindrance at the 0 degree. Okay, understood? Now, here you can see one more important thing, which is this particular peptide or polypeptide chain is composed of different planes. You can see here different planes. In fact, different planes are different peptide units, and we have seen here that each peptide unit is a planar structure, but its plane is differing from the plane of the next peptide unit, or you can say the plane of the one peptide unit is different from that of the next peptide unit or other peptide unit present in that peptide or protein molecule. Okay. And all those peptides are in different planes forming a three-dimensional structure of the protein, and you can see here that R groups are projecting out from the C alpha, and those R groups are not in the planes of those planes contributed or found by the peptide unit or peptide group. Okay. So, these orientations of the R group and orientation of one plane with respect to the another plane is possible just because of the rotation around C alpha and C bond and N to C alpha bond, and rigidity, somewhat rigidity in the structure is just because of the no rotation around the peptide bond. Okay. So, all these characters and all these attributes of the peptide bond and the unit and the bond around the peptide bond is very important to explain the three-dimensional structure of the protein, and you can say these are very important to determine the three-dimensional structure of the protein, and thereby they are also governing factors for the function of the protein, because a particular three-dimensional structure in the native condition is responsible for its function, biological function. Once their native three-dimensional structure is lost, its function is lost. So, those functions and all those factors and contributors will be discussed whenever we will study about the three-dimensional structure of the protein.
Thank you all for listening and watching the whole video with patience. All the best.